Bismillahirrahmanirrahim. Assalamu alaikum. My name is Nazia Sultan, MPhil Microbiology second semester. I am very thankful to Sir Muhammad Niaz to give me opportunity to present a topic MAXAM GILBERT method of sequencing from a subject recent trends in molecular biology. First, we understand what is sequencing. As we know that sequencing is a process combining things in exact order. Molecular biology, it is a lab process used to learn the exact sequence or order of four nucleotides or bases which make up the nucleic acid or DNA. Here we are talking about the DNA sequencing method called MAXAM-Gilbert sequencing. This process is developed by two scientists, Alan MAXAM and Walter Gilbert, in 1977. It is based on very basic understanding which is called chemical sequencing because the sequencing method rely on chemical degradation process. That's why this process is also known as chemical degradation method of DNA sequencing. It is also defined as it is a method by which the sequence of DNA fragments is identified by using chemical that cut the DNA at specific position or specific points. Let's start. Here are double-stranded DNA sample of unknown sequence and we want to sequence this double-stranded DNA by using some chemical termination process. And first step, as the sequence of both strands are unknown, but we find out the sequence of one strand, we would get to know the sequence of another one also. At first, this double-stranded DNA is denatured to a single-stranded DNA by applying high temperature or high pH. Here, the double-stranded DNA is separated into a single-stranded DNA by temperature, high temperature or sodium hydroxide. Now in the second step are on the single-stranded fragments on gel, as the lighter fragments band will move further than the heavy fragment band. But how will we know that? Which one is the lighter band? The band having larger number of purines, that is adenine and guanine, would be heavier. Here, the fragments are separated based on their molecular size. The smaller fragments move faster and will appear at the bottom, and the larger fragments will now cover more distance. And the fragments always moves from negative charge, a negative node, to the positive cathode. Now in the third step, take one of fragment band from the gel and remove the phosphate at 5 end by using an enzyme known as alkaline phosphatase enzyme. Next we add radiolabeled phosphate, that is P32, enzymatically. This is the preparatory phase for MAXAM-Gilbert method. Now we are ready to go with our reaction. Once we prepare our single-stranded DNA by taking with radiolabeled P32 at 5 end, now we start the major process, that is chemical termination process. Let's say in this process the idea is to add certain chemical agent which will cleave the single-stranded DNA fragments at certain bases. For this purpose, the reaction will occur in 4 separate test tubes. Each tube will have free agent that cleave the sample of DNA of the single-stranded at specific site. For example, we add dimethyl sulfate, high temperature plus pH by adding in combination with the sodium hydroxide, which cut the fragments at two positions, at adenine and guanine position. And test tube 2, we add dilute hydrochloric acid in combination with the dimethyl sulfate, which cut the single-stranded DNA at position adenine. Reagent hydrazine and piperidine are added to tube 3, which cut the fragment at two positions. One is thiamine and the other is cytosine. In the last tube, hydrazine, piperidine, and sodium chloride are added. That breaks the DNA fragments at position cytosine. Ultimately, DNA are broken down into number of fragments. We will get number of fragments from each tube. That is, from tube 1, we get 3 fragments because there are 2 guanine and 1 adenine, and the chemical cut the strand at these positions. From tube 2, we get 1 fragment because it cut the fragments or it break the fragment only at one position, that is at adenine. From tube 3 we get 5 fragments because there are 2 cytosine and 3 thiamine, and the chemical cuts the fragment at these 2 position bases which are present. From last tube we get 2 fragments. Once the DNA cleaved at specific residue, the fragments are separated by gel electrophoresis. And look at the data, because after separating the fragments, we will go for radioautography, which tell the position of each band labeled with P32 isotopes. As a result, we can see only the labeled fragments and interpret the result. We have 4 tubes. Each tube loaded into a separate well, and don't mix the mixture. If we mix the mixture, it will completely destroy the result. After loading the gel, the fragments start moving from negative end to the positive end, and the fragments will move according to their size. The smaller fragments move faster and will appear at the bottom. Now gel exposed to extra film to prepare autoradiography, and we detect the labeled fragments, and now we find out the sequence, which is always right from 5' to 3' end direction. The smaller fragments which are present, 2 in number at the bottom, from tube 1 is from tube 1, that is from A plus G, and the other is from tube 2, that is from the adenine tube. If the situation is like that, the base which is common in both will be present. So here adenine is present. And second label fragments present only in tube 3. So the T plus C, so the base will be thiamine, which is uncommon. Base 3 will be guanine. Same situation like base 2, that is uncommon one. Base 4 is cytosine because C is common in both tubes, that is in tube 3 and tube 4, and so on. So the sequence will be like that from 5' to 3'. That is adenine, thiamine, guanine, cytosine, guanine, thiamine, and adenine. From this strand, according to Watson and Crick model, we can identify the complementary strand. According to Watson and Crick model, adenine will always pair with thiamine, and thiamine will always couple with adenine. Cytosine will always pair with guanine, and guanine will always pair with cytosine. So the complementary strand will be like thiamine, adenine, cytosine, guanine, cytosine, adenine, thiamine. If you see the sequence on gel from 5' to 3' and compare it, it is the same sequence that we have at first. Fragment sequence that was selected, fragment sequence identified from the gel. Thank you for your attention. Allah Hafiz.